Welcome to the deep dive, where we take some really dense source material today. It's advanced texts on plant physiology, and we turn all that complexity into insights for you.
Our mission today, and it's a big one, is to give you a shortcut into the hidden universe of plant life. We're going
Yeah.
way beyond just basic photosynthesis.
Right, we're getting into their cellular architecture, their survival strategies, even the little molecular pumps that keep the whole kingdom running.
It's all about how dynamic and, honestly, how aggressive plants can be.
So it starts small, right? Deep inside the cell with genetics, then we'll zoom out to see how they manage their environment, water, energy, all of that.
And we'll finish up with growth, reproduction, and some really sophisticated defense strategies. It's a journey from the nanoscale all the way to the ecosystem.
Let's do it. So the cellular level. The thing that really jumped out at me from the sources was this idea of the simplest. I always thought of plant cells as these individual walled-off boxes.
Yeah, that's the common image. But it's not really the case. Functionally, the entire plant is one massive, unified cytoplasmic continuum.
And that's the simplest.
That is the simplest. And it's all physically connected by these tiny tubular channels. They're called plasmodesmata.
How tiny are beta?
Oh, incredibly small, about 40 to 50 nanometers wide. But think of them as high-speed communication highways.
So they're passing proteins, nucleic acids, that kind of thing back and forth.
Exactly. It's how the cells coordinate. It's critical infrastructure.
But then you have this amazing exception, which I love, the guard cells.
Ah, yes. The
Yeah.
ones that open and close the stomata, the little pores in the leaf.
Our sources say they're totally isolated, no plasmato.
They have to be. They're like a sovereign state inside the plant. If they were connected, they couldn't respond independently to signals for, balancing water loss and CO2 intake.
They need to be autonomous.
Completely autonomous. It's a perfect design.
Okay, let's zoom in even further. The sheer production capacity of these cells is just wild.
It is. I mean, you take something like the Golgi bodies or dictyosomes. A really active cell, say a meristematic cell that's dividing, can have up to 100 of them.
100. Wow. So that's the factory floor regulating all the secretions?
And for some cells, that's a huge job. Think about a root cap cell.
The one that's pushing through the soil.
Right. It's constantly pumping out this slime, mucopolysaccharides, to lubricate its path. Every time it secretes a vesicle, it's adding membrane to its surface.
So it's got a surface area problem. It would just balloon.
It would literally rupture. So to balance that, it relies heavily on membrane recycling, specifically endocytosis. It's constantly pinching off bits of its own membrane to bring them back inside.
That's incredible. Okay, speaking of surprising internal chemistry, plants have cholesterol. I never would have guessed that.
It's a great piece of trivia.
Yeah.
They do, but it's a tiny, tiny amount. Maybe 50 milligrams per kilogram of lipid.
So it's not the important part.
No, the important part is that they have hundreds of other sterols, like cetohostrols. And some of those, a group called the brassinosteroids, act as critical hormones.
Hormones, like in animals.
Exactly like in animals.
Yeah.
It's this beautiful example of evolutionary convergence. Chemically similar molecules evolving to carry these crucial regulatory signals across kingdoms.
A shared ancient language. Amazing. So moving from structure to control, how do plants manage their genes? It's more than just simple transcription, right?
Oh, much more. They have these incredibly deep regulatory layers. The first one is epigenetics.
Right, like histone methylation.
Yes, specifically on H3 lysine residues. Think of it as a master on-off switch for whole chunks of the genome.
So how does that switch work?
Well, methylation on one spot, H3 lysine 4, is generally a sign that genes are active, ready to go. But methylation on another, H3 lysine 9, usually means those genes are silenced, locked down.
Like long-term memory.
It is. But for finer, more immediate control, they have this amazing cleanup system, the RNA interference pathway, or RNAi.
This is their internal censorship office.
That's a perfect way to put it. It starts with these proteins called dicer-like that chop up RNA into tiny pieces.
And those pieces are the guides.
They're the guides. They load into this big enzyme complex called RASC and guide it to matching messenger RNAs.
And the RISC complex just destroys them.
It either cleaves them or just silences them. It's the cell's police force, deciding which genetic messages get delivered and which get shredded.
And of course, this level of control is why we've been able to manipulate them for so long.
Absolutely. The sources remind us we've been doing it for centuries. I mean, we turned wild teosinte into modern maize.
But now biotech lets us do it instantly, between species that would never cross in nature.
And that's where the modern discussion really starts. The technology offers this incredible potential for food security, but it's always a debate about balancing that against acceptable risk.
It really shows the huge implications of these tiny systems. Which brings us right to part 2, how plants manage massive physical challenges.
Yeah, like moving water. When you look at a giant tree, it's just mind-boggling how they get water from the roots all the way to the top.
And it all comes down to the cohesion tension theory.
Right. The water is literally pulled up through the xylem. It's under negative pressure tension created by evaporation from the leaves.
So it's a huge straw, basically. And the pipes themselves, the tree elements, they're actually dead cells, right? Just hollow tubes.
Dead, lignified, hollow conduits.
Yeah.
But the safety engineering in them is, well, it's remarkable, especially in conifers.
You mean the bordered pits?
Exactly. And inside those pits, there's a special structure called the torus.
Okay, so this is where it gets really clever. What does the torus do?
It's a safety valve. It protects against what's called an embolism, an air bubble that breaks the water column.
A catastrophic failure.
It would be. But if a big pressure difference happens, the torus gets sucked over and it seals the pit. It isolates the damage immediately, preventing the air bubble from spreading.
So if one pipe bursts, the plant just seals it off to save the rest of the system? That's brilliant.
And when the plant is facing a water deficit, a drought, it makes these really tough trade-offs.
The first thing to go is growth, right? Cell expansion stops.
It's the most sensitive process. Growth slows way down, which might seem counterintuitive if you're trying to find water.
But it's not.
It's not, because while shoot growth stops, that often triggers a relative increase in root elongation.
It stops growing up and puts all its energy into growing down.
It prioritizes survival. It pushes its roots deeper, searching for the water it desperately needs.
A smart, adaptive response. Okay, so moving to nutrition, how do we even know what minerals a plant needs?
The foundational technique is called solution culture. You basically grow the plant in a chemically defined liquid, like a Hoagland solution, with everything it needs.
And then you just take one thing out.
You take one thing out. And if the plant fails, you know that element is essential.
But in the real world, they don't do this alone. They outsource.
They absolutely outsource. They have this critical symbiotic relationship with mycorrhizal fungi.
Right, and there are different kinds for different jobs.
There are. You have arbuscular mycorrhizae, which are amazing at scavenging for phosphorus. And then you have ectomycorrhizae, which are experts at getting nitrogen from organic matter. Specialized teams.
Let's shift to the ultimate energy source, photosynthesis. The Z scheme, 2 photosystems.
Yes, photosystem I, or PSI, is tuned for light at 700 nanometers, and photosystem 2, PSII, for 680 nanometers. They work in series.
But they're also fragile. Too much light is a problem.
A huge problem. It's called photoinhibition. Too much light energy actually damages the system.
Sunburn for a plant cell.
That's a good way to think of it. It specifically targets the D1 protein in PSII, and the cell has to completely disassemble the whole complex to repair it, which costs a ton of energy.
So what's the quick fix? Do they have, like, cellular sunglasses?
They kind of do. In highlight, they'll actually move their chloroplast to the size of the cell, literally getting them out of direct sunlight to minimize absorption.
They physically move them out of the way. That's amazing. And what about this strange footnote on energy, the alternative oxidase pathway? It's something mammals don't have.
Right, and it's fascinating because this pathway, it bypasses parts of the normal electron transport chain.
Which means?
Which means it's less efficient. It results in a lower overall ATP yield.
Wait, so the plant is intentionally making less energy from the sugar it just worked so hard to produce. Why?
It's a trade-off. By being less efficient with energy, they can prioritize other things, like releasing that extra energy as heat to attract pollinators, or just maintaining metabolic flow under stress. It's function over fuel economy.
Like a biological sports car, that's a perfect lead into part 3, the developmental blueprint, where growth is often about, well, destroying things.
This is one of the most elegant systems in biology. Major hormones like auxin and gibberellin, they don't just turn things on. They work by triggering protein degradation.
Through the ubiquitin proteasome pathway. That sounds complicated.
Let's simplify it. In the normal state, growth is blocked by these repressor proteins, just sitting there, blocking everything.
Okay, so the brakes are on.
The brakes are on. The hormone then arrives, and it acts like a key. It activates a specific SCF ubiquitination complex.
Like SCFTIR1 for auxin?
Precisely. That complex then tags the repressor protein with ubiquitin.
Which is like a little destroy me flag.
It's a molecular destroy me flag. The tagged protein is then immediately destroyed by the 26S proteasome. The repressor is gone, the brakes are off, and the plant can grow.
So the signal is carried by destruction. That's brilliant. And this hormonal tension defines the whole plant's shape.
Absolutely. The entire architecture comes down to this antagonistic relationship between auxin and cytokinin.
Auxin comes from the shoot and flows down.
And cytokinin comes from the root and flows up. Their ratio determines everything. In tissue culture, if you have more cytokinin, you get shoots. More auxin, you get roots.
A constant push and pull that shapes the whole organism.
And that polarity is crucial, even for something as simple as making a leaf flat.
You need a top and a bottom, the adaxial and abaxial sides.
You do. And their proper juxtaposition, mediated by genes like YBE, is what allows the leaf blade to grow out into this flat, efficient solar panel instead of just a weird tube.
Okay, let's talk timing. Plants have these powerful environmental clocks, like vernalization.
The promotion of flowering by cold, it's a form of memory.
How does that work?
In a plant like Arabidopsis, the prolonged cold triggers an epigenetic change that permanently switches off a flowering repressor gene. The gene is called flowering locus C, or FLC.
So FLC is the break. Winter happens, the break is permanently released, and then it's safe to flower.
And that switch is stable for the rest of that plant's life. It remembers it's been through winter.
Once that signal is given, the race is on. And speed matters.
It's a sprint. Angiosperm pollen tubes can grow up to 2 millimeters an hour. That's 100 times faster than in gymnosperms.
Wow. And it all starts with that first contact on the stigma, the adhesion and hydration.
Right. The stigma creates this lipid-rich foot for the pollen to land on. That connection is what allows the pollen grain to hydrate and start that explosive growth.
And finally, fruit ripening, especially in climacteric fruits like tomatoes.
Right. Those require a massive surge of the gas ethylene, plus a huge spike in respiration to ripen properly.
And we know this for sure because of genetic engineering, right?
Exactly. The proof came from transgenic tomatoes, where scientists block the enzymes that make ethylene. Those tomatoes simply would not ripen.
Unless you sprayed them with ethylene gas from the outside.
Then they'd ripen perfectly. It was the definitive proof.
Which leads us right into our last part, defense and acclimation. Because being stationary means you have to be ready for anything.
Anything and everything. The first line of defense is always physical. You've got thorns, which are modified branches. You have spines, which are modified leaves.
And then the little hairs, the trichomes.
Some of which are glandular. They're like little poison sacs. They burst on contact and release sticky, nasty secondary metabolites like terpenoids.
An immediate deterrent. But the chemical warfare gets much deeper.
Oh yeah. A classic example is cyanogenic glycosides, like the durin in sorghum.
They're harmless at first.
Totally inert, but when an insect chews the leaf, the tissue damage causes them to break down and release hydrogen cyanide gas, HCN.
A potent poison. What does it target?
It's a metabolic poison. It shuts down cytochrome oxidase in the mitochondria, which effectively stops cellular respiration cold. It's a metabolic shutdown for the attacker.
But beyond just defending themselves, they actually call for help. This is where it gets really interesting.
This is the plant calling in air support. When a grasshopper chews on a maize leaf, its saliva contains a compound called velicitin.
The plant recognizes that compound.
It recognizes it as an attack signal, and in response, it releases a cocktail of volatile organic compounds, VOCs, into the air.
And what do those VOCs do?
They're a very specific signal that attract parasitic wasps. The wasps fly in, find the caterpillar, lay their eggs in it, and kill the pest.
The plant is hiring a bodyguard. That's phenomenal. And this signaling is fast, right? It's not just chemical.
No, it's also electrical. The sources describe these rapid, long-distance electrical signals mediated by glutamate receptor-like ion channels.
So it's like a plant nervous system. A damaged signal shoots through the plant.
Exactly. And it triggers defenses like jasmonic acid production in distant leaves way faster than a chemical could diffuse there.
And this systemic response, it also works for things like heat and cold.
Yes, that's called systemic acquired acclimation, or SAA. It's mediated by a self-propagating wave of reactive oxygen species, ROS.
So a wave of ROS travels through the
A
plant.
wave of ROS, yeah. It moves at about 8.4 centimeters a minute, priming distant parts of the plant for the stress that's coming. They anticipate the danger.
Anticipation must be key for something like freezing, and the damage mechanism there is surprising. It's not the ice crushing the cells.
No, not directly. The primary injury is from ice forming outside the cells, in the apoplast. That extracellular ice lowers the water potential.
So water gets sucked out of the cells.
It rushes out, causing catastrophic dehydration. The cell dies of thirst, not from being crushed. It's essentially drought stress caused by cold.
And on the flip side, What about too much water, flooding?
They adapt by creating parenchyma. There are basically gas-filled tunnels like snorkels that form in the submerged roots and stems.
Providing a pathway for oxygen.
A low-resistance pathway for oxygen to diffuse from the leaves all the way down to the drowning roots.
This has been an incredible journey, from those tiny plasmodesmata and the elegance of ubiquitin, all the way up to these vast communication networks using VOCs and electrical signals.
It really shows that plant life is this continuous, incredibly fine-tuned process of growth, defense, and adaptation. It's a constant balancing act.
We want to leave you with a thought from the scientist Vannevar Bush. He said, "Science has a simple faith which transcends utility. It is the faith that it is the privilege of man to learn to understand and that this is his mission."
And you've just gained a huge shortcut to understanding this hidden universe.
So consider this. The soil you walk on, the trees you see, they are all running on systems this complex. Self-sealing water pipes, chemical air support, a literal nervous system. What other complexities running right under your feet have you been taking for granted?
Thank you for joining us for this deep dive.
We'll catch you next time.